Hello, and welcome to the Guthy-Jackson Charitable Foundation Education Center and the NMOSD Grand Rounds series. We welcome all in attendance and remind everyone that the Guthy-Jackson Charitable Foundation is proud to be a source of information about NMO and related conditions. The content of the following presentation is based entirely on published data and expert opinion. GJCF is not a clinical provider. It does not offer or represent diagnostic, therapeutic, or clinical care recommendation or advice on personal health. All patients and caregivers are advised to consult with their healthcare team for specific information. The NMOSD Grand Round series has several overarching goals. Each episode is designed to offer a practical summary of latest information on a specific topic. It is designed to emphasize key themes and clinical pearls. It is intentionally inclusive of non-specialist audiences. And finally, a pocket guide is included at the end of the presentation that we hope serves as a helpful reference. This presentation qualifies as a continuing medical education activity. To receive credit, please visit the GJCF Education Center using the QR code shown here. Select the session of interest and register for that session. View the session and fulfill the CME requirements and receive the CME certification online. My name is Megan Bainey, and I'm the Director of Research Programs and Contracts for the Guthy-Jackson Charitable Foundation. I'm delighted to welcome our guest speaker, Dr. Michael Yeaman. Dr. Yeaman is a distinguished professor of medicine at UCLA, chief of molecular medicine at Harbor UCLA Medical Center, and an advisor to the Guthy-Jackson Charitable Foundation. Dr. Yeaman, thank you so much for your presentation today. We look forward to learning more about emerging tolerization technologies and clinical trials.
Megan, thank you so much. It's really a pleasure to be here and, of course, to work with you, the GJCF team. But more importantly, hopefully we can update everyone on emerging tolerization technologies and clinical trials. And it's a very exciting time in the field, and I hope this presentation is helpful. In terms of the actual content, as mentioned, it is all based on information that's public domain. It's published and available per references that will be used throughout the presentation. Here are my disclosures. What I would like to do today is provided in this overview. First, a brief update on what I like to think of as the secret pandemic of autoimmune disease. Also, talk a little bit about the reason that tolerization might be so important for the future of autoimmune therapies. And that has to do with the vicious cycle of immune-modifying therapies, infection, and cancer. Then we'll get into the real interesting part of the talk, which is the unsolved mystery of immune tolerance and what is being done to solve and be able to intervene in tolerance for benefit of patients. We'll talk a little bit about autoimmune models and how NMO and MOGAD are very high on the list of diseases that really fulfill all of the checklist needed for tolerization. And then we'll go right into what I call autoimmune panacea, that is retolerizing to arrest and perhaps cure autoimmune disease. And then we move into specific emerging tolerization technologies, and then an update on clinical trials in progress and a bit of a prospectus. So with that, let us dive into autoimmune diseases as an overall impact on health in the world today. There are more than 100 distinct autoimmune diseases known. This represents almost 10% of the population. That equates to about 24 million individuals, or 640 million individuals worldwide directly impacted by autoimmune diseases. The prevalence overall of autoimmune disease is equal to heart disease and twice that of cancer. Very unfortunately is this reciprocal relationship among autoimmune disease, cancer therapy, and infection. So I'll talk more about that. There is a female propensity, and in many autoimmune diseases, it's very high, in terms of the frequency of autoimmune disease in females versus males. This likely suggests an infectious disease trade-off, which we won't be able to get into today because of time, but it really becomes important when we think about autoimmune diseases of the central nervous system, which are really special cases that deserve special attention. And this is why NMOSD, namely NMO, MOGAD, for example, now different diseases, really represent model targets for tolerization cures. And to achieve that, we need pioneering immune tolerization efforts to address these unmet needs. Very briefly, this illustration shows you the increased incidence of many different autoimmune diseases over the last nearly 40 years. The idea is that if you look at most of the lines here, you can see that very many of them increase. And in particular, there are sharp increases in many of the autoimmune diseases that impact a lot of individuals. I'll just point out an interesting theme that has emerged, and that is neuromyelitis optica, about 20 years ago, was thought to have roughly 1 in 1 million prevalence. We know today it's more like 5 in 100,000. Now, we realize that there hasn't been a 50-fold increase in actual cases, but there has been increased detection, diagnosis of NMO, such that there are now about 50-fold more people directly affected by NMO than just 25 or 30 years ago. And you can see overall many of the autoimmune diseases shown here: IBD, celiac disease, multiple sclerosis, type 1 diabetes, psoriasis, lupus, just as examples, have all increased considerably, such that the overall prevalence increase in autoimmune disease is more than twofold in the last 40 years. Well, what I want to talk about now is the vicious cycle of autoimmune therapies. And I want to be very clear that the advent of approved therapies for NMO is one of the most exciting and frankly remarkable stories in the field of neurology. So the idea is that these approved therapies are great first steps. They have saved and improved many lives. And we are very grateful to all who participated in the clinical trials, of course, the patients, companies, et cetera, to make those approved therapies a reality. We also recognize that there's room for improvement. Many of the therapies used in autoimmune diseases create infusion or injection reactions or allergies. There can be increased rates or severity of infections, including some serious infections. Some autoimmune disease therapies increase risks of cancer due to impaired immune surveillance. There can be logistics issues, of course, for example, scheduling, dosing, scheduling, laboratory monitoring, checkups, et cetera. And finally, cost burdens of therapies and insurance are not trivial. So the approved therapies are remarkable, positive steps forward. And what we're really talking about in this discussion today is the next generation beyond therapies. Here is just a brief summary of the idea behind the vicious cycle of autoimmune disease. So let's imagine a patient is diagnosed with an autoimmune disease, and they are placed on immune suppressing therapy to control their disease. Well, many of these therapies increase risks of infection, and infections in the US and worldwide are increasingly resistant to antimicrobial therapies. In addition, a large number of cancers are due to infections that are otherwise preventable. So now you have a bit of an insight into the idea that immune suppression can not only lead to infection, but can potentially lead to cancers. And cancers then are treated oftentimes, and increasingly so, with checkpoint inhibitors that unfortunately can cause autoimmune disease. So it's this vicious cycle, and we really need to break this cycle going forward. So this is a theme that I think you'll appreciate in terms of why tolerization. Well, let's now move to the unsolved mystery of immune tolerance. And I want to try to bring everybody to the same sort of level on the immune tolerance and the immune system as it relates to tolerance. And so let's just start from the beginning in a basic way, what is immune tolerance? And really, in a nutshell, immune tolerance is how your immune system protects you against internal or external threats, such as cancer or infection, without harming any molecules, cells, tissues that make up the body. Put another way, it's a system of checks and balances that prevents the immune system from targeting healthy cells and tissues. Well, immune tolerance, and in fact, immunity overall, is not a static process. It's constantly reacting, turning on, turning off, turning up, turning down. And if you think about it in this sense, as illustrated here, on the right side are many of the healthy requirements of an immune system to allow development and growth, to allow wound repair and remodeling, to allow a healthy microbiome, and to allow healthy pregnancy. And on the other side is sort of the adverse potential for the immune system. For example, if it misdetects or misrecognizes transform cells and allows neoplasias and cancers, those are bad. Of course, infections and environmental exposures can be a problem, and the immune system needs to defend against those. And unfortunately, the immune system can cause or contribute to miscarriage, graft rejection, and other problems. So there are endogenous, that is signals from within that the immune system has to monitor and respond to, and there are signals from the environment that it must respond to. And if you think about it in this way, the immune system is constantly acting as a gyroscope to balance, benefit, and defend against risk. Well, very briefly, the immune system, as it relates to tolerance, emerges from the bone marrow. Like most immune cells, T cells develop in the bone marrow as immature cells. They traffic to the thymus, and the thymus has three components that are sort of different microenvironments. The capsule, further into the gland is the cortex, and the deepest part of the thymus is called the medulla. So T cells made in the bone marrow traffic to the thymus and undergo maturation in each of these places in the thymus. So going from the cortex to the medulla, there are three steps that T cells must fulfill in order to become potentially mature in immune tolerance. The first, can the T cell recognize antigen presentation in context of MHC by an antigen presenting cell? And I'll talk more about that in just a second. If the answer is yes, they move on. If the answer is no, they are deleted. The second exam, if you will, that T cells undergo is, can they detect the primary, secondary, and tertiary signals in exchange with antigen-presenting cells? And we'll talk more about those in just a moment. If the answer is yes, they move on. If the answer is no, they are deleted. And finally, can the T cell appropriately engage the autoantigen and its epitopes by way of binding affinity and other factors? And if the answer is yes, the T cell moves on. If the answer is no, they're deleted. Less than 10% of all T cells graduate from the thymus. Here's a more detailed sort of summary of what I just mentioned. So you have a naive T cell here. This is a medullary thymic T cell, and they are exposed to antigens by way of antigen-presenting cells. These are professional antigen-presenting cells, like a thymic antigen-presenting cell, a thymic dendritic cell, or by way of B cells. In some cases, there can be cooperation between these two cells for antigen presentation. In the case of antigen-presenting cells, there are specific gene regulatory aspects that mediate expression and detection. Those include AIR and FEZF2. And then BCRs, the B cell receptor diversification, is involved in presentation of antigens to T cells. In either case, if there's inappropriate response to these antigen presentations, T cells can either be censored by programmed cell death, or they can be edited and or energized by way of the T cell receptor. So in either case, these T cells no longer function and are removed. The T cells that are allowed to move forward typically are what we call restricted, either to MHC2, which would be a CD4 type of T cell, MHC1, which would be like a CD8 type of T cell. But there are also T cells that emerge that are regulatory T cells. These are cells that put the brakes on the immune system in response to specific antigens. So you have effector cells and regulatory cells. Well, once graduate, these cells move from the thymus into lymph nodes. And you can see lymph nodes are located in sort of intensified regions throughout the body. They include cervical lymph nodes, axillary lymph nodes, etc. And when T cells move to these lymph nodes, they basically wait here for antigen-presenting cells in the periphery to bring them antigens that they have seen in the periphery. They expose the antigens and the epitopes to the T cells in the node, and those T cells are either activated or inactivated. The activated T cells then emerge from the node and move toward the site of the threat, be it cancer, infection, etc. And that is shown here in terms of how the interactions in the node occur. So antigen-presenting cells will interact with naive T cells that have graduated from the thymus. There is a bidirectional handshake, if you will, by way of the antigen-presenting cell showing the antigen to the T cell, but then it also needs a counter response, and I'll talk about that in just a moment. Similarly, B cells need to have cognate approval from T cells in order to mature. And in either case, there can be anergy, where the T cell is inactivated, or programmed cell death, apoptosis, where the T cell is deleted. Nonetheless, effector cells can emerge. They are antigen specific at this point. And there are also regulatory T cells that emerge. They can either be Treg cells or TR1 cells. There are a few other kinds of regulatory T cells, but these cells prevent overactive effector T cell functions. So what causes loss of immune tolerance? Well, in a nutshell, there typically is a change in the antigen. This could be structurally, it could be post-translationally, it might be where the protein or its antigens are expressed. So these include genetic, epigenetic, and ectopic causes of loss of immune tolerance. There can be inappropriate antigen presentation by way of the HLA or the MHC system. There can be dysfunctional T or B cell genes that encode receptors. And of course, inappropriate or misfunctional T or B cell receptors can lead to loss of immune tolerance. Those receptors and their recombination are encoded by the recombinase activated gene system. Many people believe that infectious diseases and/or environmental antigens are largely to blame for loss of immune tolerance. If so, that would be due to something called antigenic memory, where, let's say, a pathogen has a protein that looks similar to a human protein, and the immune system simply makes a mistaken identity. There can also be causes of lost immune tolerance by way of pregnancy, cancers, and neoplasias, and other types of graft transplantation. So these introduce neoantigens, and the immune system may make mistakes. Well, let's think now about NMO and MOGAD as models for tolerization. And I'll just summarize sort of what we know about the science of NMO and MOGAD in a way that hopefully will set the stage for what we'll talk about afterwards, which is how to intervene in these steps. And I've gone through this earlier in the talk, but just to reiterate, so this, in the case of NMO, is an astrocyte. It's expressing aquaporin 4 on its surface, and I'll focus on NMO as an example, but many of these same themes occur in MOGAD, where this cell would be an oligodendrocyte, and the protein would be MOG protein. But let's say this is NMO, here's aquaporin 4. For reasons that we do not fully understand, antigen-presenting cells will see this cell and its aquaporin 4 protein. It will process those proteins and put pieces of them on its surface. This is called antigen presentation. And then we have this bidirectional molecular handshake. The epitope of aquaporin 4 is presented to the T cell receptor. That's called signal 1. The T cell in turn counter handshakes with the antigen presenting cell. That's called signal 2. And all of this occurs in context of cytokines, which we think of as signal 3. And these cytokines sort of add a degree of urgency or indifference to this antigen presenting scenario. But when autoreactive T cells encounter epitopes presented by antigen-presenting cells, these autoreactive T cells now are activated to respond to our own proteins, and they can be polarized. In the case of NMO, it's polarized to Th17, and these are now autoreactive effector T cells that can really cause a lot of pathogenic steps in NMO. Same thing for MOGAD, we believe. We just know a little bit more about the science of NMO, at least as it regards tolerization. Once the autoreactive T cell is now fully armed and able, it will interact with naive and other types of B cells. Again, there's a bidirectional handshake here. Autoreactive T cells are required to approve B cells to make antibody. And of course, in NMO, these antibodies are targeting aquaporin 4. So they move through the blood-brain barrier, bind to aquaporin 4 on the astrocyte in the central nervous system. Whenever antibodies of certain types bind to proteins, they can activate something called classical complement fixation. One of the key proteins in classical complement fixation is C5. It's cleaved to C5a, which diffuses back into the periphery and activates neutrophils, eosinophils, and macrophages, which then move by way of margination and diapedesis into the central nervous system and intensify as an inflammatory cell population that can cause further inflammation and demyelination in the central nervous system. So we need to consider all these steps with respect to how do we intervene by way of tolerization. So why NMO and MOGAD are very good tolerization models, and again, I'll just use NMO as the example here. There is an established immunodominant autoantigen. In the case of NMO, it's aquaporin 4. In the case of MOGAD, it's MOG protein. Autoimmune mechanisms and epitopes are fairly well understood. The inflammatory demyelination pathogenesis is pretty well understood, as are the neurological deficits that lead to defined clinical phenotypes. And one of the emerging advances in the field is a relapse definition that is being harmonized by way of an international clinical consortium. Both diseases have increasing prevalence, likely because of increased detection and diagnosis. Both of them can have catastrophic outcomes. There are 4 approved drugs, of course, a remarkable advance, and there can still be relapses in some instances and some serious adverse events which make tolerization attractive. And finally, there's strong clinical, industry, and patient interest in tolerization, particularly for patients who are not well managed or controlled by the approved therapies. All right, well, if in theory tolerization is possible for autoimmune diseases, it would be a panacea, because if we can solve one autoimmune disease, we should be able to solve them all using the immune system in the same way, and that is re-educating it to not consider autoantigens as foreign or a threat. So what then is immune tolerization? Well, it's restoration of immune tolerance to self-antigens that would otherwise cause autoimmune disease, and by doing so, arrest and potentially reverse autoimmune pathogenesis. Very importantly, tolerization is unlike therapies currently because it targets and uses the immune system in a dynamic way to reprogram the immune system to be tolerant to the specific autoantigens, but not affect the ability of the immune system to protect against cancer or infection. So it spares host defense against bad things, and it enables host tolerance against otherwise normal tissue. A special case for tolerization is what we like to think of as curative immune tolerization. And if this is possible, and we think it is, this would reset the cellular and molecular mechanisms of immune tolerance in a manner that encodes tolerogenic immune memory. which means not only would there be tolerance based on the tolerizing therapy, but the immune system would learn and remember not to attack self-proteins like aquaporin 4 or MOG. And some of those types of tolerization and immune memory mechanisms can last a lifetime. We'll talk more about that in a second. Let's also differentiate between desensitization and tolerization. So desensitization has been around for a long time. It's used often in allergies. So typically allergies are caused by exogenous stimuli like pollen or plant extracts, and these cause typical allergies. In tolerization, we're really talking about an endogenous stimulus like an autoantigen, like one of our own proteins or tissues, and that's associated with autoimmunity. In desensitization, the approach is to use escalating doses of the allergen itself, and that's done in a holistic way and typically leads to benefit. With tolerization, we need to contextually expose immune cells to autoantigens in a way that reprograms them. And we've talked about that a little bit. We'll come back to it in just a moment. The goal with desensitization is increase allergen reaction threshold. That is to make an immune system less and less interested in responding. But that only lasts as long as you do the therapy. In tolerization, we want to induce autoantigen-specific non-response. That's really what we're interested in. So whereas desensitization will skew T cells to a Th0 polarization schema and shift B cells from IgE, which typically is related to allergies, to more of an IgG profile, and particularly an IgG4 subset profile if possible. Whereas tolerization is more cell-driven in the sense that it really intends to identify and induce regulatory T cells and exhausted T cells that would otherwise react to specific autoantigens. Typically, there aren't a lot of cellular laboratory changes in desensitization, more molecular, but there are large-scale changes to autoreactive T and B cells anticipated in tolerization. And a key point here is desensitization is temporary. It only lasts as long as you give the therapy. Whereas what we are hopeful for is that immune tolerance can be restored in a way that it goes beyond exposure to therapy and lasts a much longer period of time. How might this be done? Well, basically, it's correcting the causes of lost immune tolerance. So we would want to correct gene mutations or epigenetic modifications or ectopic locations of gene expression that lead to autoantigen detection. That might occur including ways that correct protein production or structure. By way of a process called immune deviation, we can reprioritize the immune responses, let's say from cellular to antibody or from antibody to cytokine, so that the process of autoimmunity is fundamentally altered. Of course, there can be deletion of autoreactive antigen-presenting cells, T cells or B cells, and that's called censoring. And finally, the idea that was most attractive is to basically reprogram the immune system so it no longer thinks of the autoantigens as foreign. And as I mentioned, that involves potentially autoreactive antigen-presenting cells, T cells and B cells, and turning them into regulatory or non-reactive immune cells. Well, we know that there's a lot of clues to NMO tolerization already in the literature, and we've been working very hard over the last 15 years or more to turn this science into pathways to tolerization. So for example, we know that in some patients who have anti-aquaporin 4 NMO, there are potentially protective antibodies that emerge. And if that's true, then we know the immune system is already learning on its own how to autocorrect in those patients. There can be certain subsets of cells that reflect autoreactive T cell repertoires that might be beneficial as compared to harmful. Autoreactive B cells may serve as alternates for antigen presentation, and if that's the case, then those might be targeted in a different way. Recent studies by the team at Northwestern have shown that if you knock out interferon gamma in mice, there is a strong propensity for IL-17 driven NMO. And this just speaks to the balancing act between certain types of tolerization. And I'll move right to that, for example, where if we're thinking about pathways to tolerization, we really want to talk about non-exclusive pathways. So what I've shown here are three different avenues to tolerization. One is to disrupt antigen-specific activation. One is to induce antigen-specific regulation. And one is to impair antigen-specific effector functions. And the whole point to this slide is that one or more pathways can be used at the same time for tolerogenesis. Okay, let's talk about the technologies that are emerging to actually cause restoration of immune tolerance, many of which have extensive publication in the literature and are now being moved from the laboratory to the clinic. In a nutshell, this illustration basically shows the general categories of technologies for antigen-specific tolerization. They include CAR T and AA CAR T cell therapy. I'll talk about that. This is a special case which really is more about cell depletion as compared to cell reprogramming, but we'll talk a little bit about that. There can be antigen-specific tolerogens. These are artificial epitopes and pieces of proteins that, rather than causing inflammation, can be used to cause the immune system to become tame to these proteins. Likewise, antigen-specific tolerizing nanoparticles contain pieces of the autoantigen, but in a nanoparticle that causes the immune system to ignore or to become tolerant of these proteins. Dendritic cell vaccines. These are antigen-presenting cells where you feed the dendritic cell pieces of the autoantigen, but you teach the cell to be tolerant of those proteins as you're feeding it to them. Interestingly, the opposite of treatments in cancer. So in treatments in cancer use checkpoint inhibition. In autoimmunity, we want checkpoint induction. And if we can do that in an antigen-specific way, that's a great method for tolerization. There are also very interesting studies going on now using HLA tolerogenesis. This is the special type of antigen presentation that occurs in a fetus where the mother and the fetus immune systems learn to get along. And that is a very interesting approach to potential autoimmune tolerization. There are inverse nucleic acid vaccines where the particle containing the piece of nucleic acid encodes an autoantigen, but it encodes that autoantigen so that it is produced in a situation where it presents the immune system a reason to accept the protein encoded by this nucleic acid as self and not to be attacked. There are some interesting technologies that move certain types of genetic information into autoreactive cells, including autoreactive antigen-presenting cells, such that the cell recognizes the autoantigen and undergoes programmed cell death. So that's a two-in-one kind of an approach, which is very interesting. I won't say much about endogenous tolerogenesis, except, every one of our red blood cells is detected by our immune system and removed at the appropriate time. Certain technologies are trying to use that platform to tell red cells and the immune system that autoantigens associated with red blood cells can also be tolerated. And finally, the most extreme technology for tolerization is autologous hematopoietic stem cell transplant or bone marrow transplant. Of course, this is basically replacing the immune system in a patient. It's more intensive, but there have been early studies that suggest promise. Long-term efficacy, we're still waiting to see how that works out in the long run. Very briefly now, just a rundown on some of how these tolerization technologies work. In the case of CAR T or AA CAR T cell therapies, healthy T cells are removed from a patient, that is non-autoreactive T cells. In the laboratory, they are engineered by way of DNA transfection, to encode receptors for particular antigens that are specific to a disease. These T cells now recognize and destroy cells that have that antigen on them. So that could be T cells or it could be B cells. For example, CD19 B cells can be destroyed by certain types of CAR T therapies. In terms of lipid nanoparticle tolerogenesis, again, these particles have pieces of autoantigen in them. They're fed to antigen-presenting cells in a way that makes these tolerogenic dendritic cells or tolerogenic antigen-presenting cells. They're now able to go and teach the immune system, rather than respond to the autoantigen of interest in an inflammatory way, let's respond to those autoantigens in regulatory ways that are non-inflammatory. Very similarly, dendritic cell vaccines, where dendritic cells from patients are removed by way of the bloodstream, they're exposed to the autoantigen epitopes such that they become tolerogenic by way of certain types of conditions, dexamethasone, TGF-beta, some other exposures. These tolerogenic dendritic cells are now programmed to induce tolerance to the specific autoantigens. They're returned to the patients and are felt to induce tolerogenic B cells and T cells specific to those antigens. Inverse RNA or DNA vaccines, again, the idea is to put pieces of nucleic acid that encode an autoantigen into a very special type of nanoparticle. And these can be nanoparticles that contain special modifications of mRNA, like pseudomethyluridine, or specific types of lipid nanoparticles that induce tolerance of dendritic cells by way of increased PD-L1, CTLA-4, or other checkpoints, and then go on to induce regulatory T and B cells in the patient. And then finally, in these examples, is the programmed cell death or dendritic cell tolerogenic vaccines, where DNA encoding the antigen target gene, or in this case, the autoantigen target, as well as mediators of programmed cell death, so that the autoreactive dendritic cell is then deleted or made into a regulatory subtype. So these are just a quick rundown on the types of technologies. And now let's briefly review some special considerations for clinical trials in tolerization as we move to the conclusion of this presentation. First of all, there are very important clinical tolerization endpoints. The first, safety. And this is an acceptable profile of acute response so that there's not too much anaphylaxis or cytokine storm. And most importantly, no new disease activity, no symptoms, relapses, no new comorbidities. Of course, efficacy. Is the tolerogenic therapy or tolerization regimen able to benefit patients by way of changing biomarkers, by changing clinical symptoms, by impeding or eliminating disabilities, by way of relapse prevention, etc. We're also very interested in how long the tolerogenic effects last. Is it durable? Does it need to be given in a boost from time to time, or can it be potentially lifelong? And then finally, all of this work needs to understand what are the biomarkers of tolerization? What molecules change? What cells change? What imaging or other correlates might be linked to benefit by less inflammation and more immune regulatory mechanisms? If we think about when tolerization might be best, this is sort of a very high-level overview going from predisposition, which might include genetics, latent viruses, family history, by way of endogenous stimuli or exogenous stimuli, for example, birthing route, exposure risks, infection, etc., moving into sensitization, where perhaps through epigenomic changes, neoantigens, microchimerism, or other reasons, or by way of exposures, behavioral risks, etc., a person is sensitized, increased propensity for autoimmune disease, but it has not yet manifest. Next would be the pathogenesis step, where actual tolerance is lost by way of autoreactive T and B cells. Pathogenesis begins, and you can start to see various types of manifestations, which ultimately leads to clinical disease. Interestingly, clinical disease is often where the current therapies target, that is, preventing relapses, for instance. In tolerization, we really want to move this all the way forward to before pathogenesis even becomes a problem. So we want to reach upstream, retrain, reprogram the immune system to prevent pathogenesis wherever possible. There are special considerations in terms of what is called immune induction in some types of tolerogenic therapies. For example, in CAR T cell therapy or in bone marrow transplant, you have to condition the immune system to accept a graft. That can come at some risk and some types of responses that we just have to carefully monitor, but in general are well managed. So there are different kinds of induction regimens. These can include blocking the molecular handshake that I talked about. They can include depleting certain subsets of cells that participate in disease pathogenesis, like B cells, or they can include both. For example, a cell inhibition type of regimen, including cyclophosphamide and filgrastim, but also something like rituximab. So you get sort of a bimodal type of immune induction. We have learned a number of things from tolerization clinical trials that have been ongoing for some time now. First, the importance of specific clinical endpoints. Without specific clinical endpoints, equipoise rules. And that's really the enemy of a clinical trial. So we really need to have specific clinical endpoints. The idea of targeting autoreactive T cells or B cells is very difficult historically. Finding these autoreactive T and B cells has been tough, but there are now advances in immunologic techniques that can help us find them. The absence of molecular and cellular biomarkers has been a problem in clinical trial outcomes to date. I mentioned immune induction and how it may create confounding issues in and of itself. There is a challenge between autologous therapies, which are, for example, cells that come from the patient being treated, versus allogeneic, which are cells that come from patients or persons not being treated, and how manufacturing and FDA review of some of these cells is challenging. And finally, there can be, in some historical examples, new disease activation despite preclinical safety profiles. So clinical trials have to be done very carefully, safety first. Here's an example of how a clinical trial for tolerization might be done in NMO or MOGAD. And this stems from a consensus working group of one of the Guthy-Jackson Charitable Foundation summits for NMO tolerization. The idea is no patient should be left in a placebo, untreated exposure window. And how might that work? Well, for example, patients can be randomized into two arms, standard of care or standard of care plus the experimental tolerization regimen. As time goes on, only patients in the experimental arm that exhibit tolerogenic biomarkers would begin a process of tapering the standard of care. That would leave them on the experimental regimen and allowed to fulfill the time remaining in the study. Meanwhile, in parallel, the control individuals would be remaining on ongoing standard of care. So you can see this is a way to include tolerization therapy and in particular, allow it to be on its own in a patient only when the patient exhibits tolerogenic profiles. All right, very briefly, let's go through an example and then a little bit of a high-level review of where things are in clinical trials for tolerization in NMO and MOGAD. And I'll just use this example, which is now a couple of years in publication of a therapy called ecocapigene autoleucel. And this is a cell therapy. This is a CAR T cell therapy that targets the B cell maturation antigen, also known as the BAFF ligand. So I won't go through the details of this procedure, but this is an autologous CAR T cell therapy. It was tested in 12 patients, a median age of 49 years, 83% were female. These patients were anti-aquaporin 4 positive, diagnosed with NMO. Their annual relapse rate was significant. It was greater than or equal to 2, and they had disease which was refractory to other therapies. In the delivery of the CAR T cell therapy itself, all the patients experienced grade 3 adverse events. This means that, as in all CAR T cell therapies, there's typically a hospitalization process which allows the patients to be carefully monitored, because we know some of these approaches will cause potential immune over responses and possibly infections. And a grade 3 adverse event relates to, there was a pretty big drop in the types of immune cells that were being monitored. There was no major cytokine storm response. There were infections, including UTIs and re-emergence of some latent viral pathogens, including cytomegalovirus. There was no PML. No patient needed tocilizumab to control cytokine response reactions. The results were quite promising. Whereas prior to the advent of the CAR T cell therapy in these patients, there were lots of relapses. No relapse occurred in 11 of 12 patients that continued on the study, and this went up to 14 months. It's now a couple of years. The data have shown some relapse events, but overall pretty efficacious. Importantly, the mean anti-aquaporin 4 titer went from 1 to 1,400 to 1 in 5 after therapy. So the antibody titer dropped dramatically. The disability score dropped from 7 to 4.5. And as a result, a company in China has filed a BLA for this therapy, which is now being moved into clinical development. So lastly, I just wanted to give you a sense of how fast the field is accelerating. So whereas 15 or 17, 18 years ago, the question was, is NMOSD tolerance even possible? Well, I'll just bring your attention to the number of studies that have strongly supported the answer to that question, is that it is not just possible, but it's probable, ranging from early bone marrow transplant to tolerogenic dendritic cell vaccines in NMOSD, to anti-MOG inverse vaccines for EAE, which is a MOG model, to lipid nanoparticles that have looked very promising in other autoimmune diseases like celiac disease, to B-cell CD40 tolerization in NMOSD, and then as recently as this year, a tolerogenic lipid nanoparticle mRNA inverse vaccine for MOG. At the same time, there are a number of CAR T cell therapies that have been moving quickly forward. These range from targeting CD19 B cells in lupus to, as I just mentioned, the B cell maturation antigen in NMOSD. And there are now bispecific CAR T cell therapies being evaluated in MS. There are other fast-track CAR T cell therapies that evoke checkpoint inhibitors. And then most interestingly are these universal bi or tri-specific CAR T cell therapies that are really moving quickly into clinical evaluation. So there's a lot of promising activity going on in the field. I'll just stop there and end with the clinical pearls. And I probably summarize it like this. Immune tolerance is a dynamic process involving central or thymic tolerance as well as peripheral tolerance that are largely driven by antigen-presenting cells, T cells, and B cells. Loss of immune tolerance is unclear, but it likely involves genetic, epigenetic, infectious, and or perineoplastic mechanisms that are very likely to be solved as we move forward. The goal of curative tolerization is to permanently restore immune tolerance by creating regulatory immune memory. We want the immune system to be tamed and to remember that it should not think of aquaporin 4 or MOG protein as an enemy. And as I just showed you, there has been a dramatic surge in tolerization technology development and clinical trials with promising early success in NMO and MOGAD and related conditions. And finally, I'll just leave you with this pocket guide. This is just a quick reference guide to the technologies that I discussed, which types of cells they target, a general mechanism by which they're believed to work. I did point out that different types of induction are required for different approaches here, the most intensive of which are for CAR T-cell therapy and bone marrow transplant, where in general these technologies are staged in clinical trials, and whether there's proof of concept in NMO, MOGAD, or NMOSD. So with that, Megan, I'll just leave people with a couple of thoughts for the future. The first is tolerization does not ask an immune system to cease its functions or wait on the sidelines. Rather, as illustrated by this really interesting piece of art by Banksy called Rage, the Flower Thrower, tolerization asks the immune system to actively participate in a new process of peace. And finally, I'll just offer one thought about the probability or the possibility of inducing antigen-specific immune tolerance as a cure for NMO, MOGAD, and other autoimmune diseases. It is a bold, ambitious goal. But I'll just use Christine Peterson's words here. She was one of the first women in the NASA program. And she said, if one looks into the future and an idea today seems like science fiction, it may be wrong. But if it does not look like science fiction, it will definitely be wrong. So I'll just leave you with that idea of, yes, it's a bold, ambitious goal to cure autoimmune diseases, but we think it can be done, and we're working hard to do that. Let me turn it back over to you, Megan, and thank you so much for the opportunity to present here.
Dr. Gaiman, thank you so much for an excellent presentation and overview of emerging tolerization technologies and clinical trials in NMOSD. We have a few minutes for some questions. Let's get started with the first one. Is CAR T therapy considered tolerization or immune cell depletion?
Thanks, Megan. It's a very interesting question. So technically, it's immune cell depletion because CAR T cells seek and destroy cells that they have been engineered to find. That said, there's really interesting after effects of CAR T cell therapy that suggest they have a much longer lasting impact on tolerance than we had foreseen. So I would call it immune cell depletion, but it's possible that there might be tolerogenesis occurring in ways that we don't fully understand yet.
Thank you so much. For our next question, is laboratory monitoring required in follow-up for tolerization therapy?
So certainly for more intensive tolerization therapies, but actually for all tolerization therapies, there will be close monitoring of cells and cytokines so that we know, what's actually happening to your immune system. Is it becoming more tolerant to these autoantigens? What cells and cell subsets are changing? What cytokines are changing? How does this offer insights into polarization of the immune system, which hopefully will lead to more of a regulatory polarity? So yeah, there are going to be lab monitoring, checkups and that sort of thing, but it's really an important part of the process. We learn more from that.
Excellent. Thank you, Dr. Yeaman. Let's go to question number 3. Do tolerization clinical trials involve untreated placebo control groups?
So this is a very key question. Prior to the advent of approved therapies, tolerization trials would probably require placebo. But now that there are approved therapies as standard of care, for example, it is very unlikely. In fact, it would probably not be allowed by the FDA to perform a tolerization clinical trial using an untreated control group. So as I mentioned in that model clinical trial, the idea would be to have patients on standard of care or standard of care plus tolerization. And when the patients on tolerization therapy exhibited biomarkers of tolerogenesis, they would remain on the tolerization therapy, but their standard of care therapy could be tapered. And in this way, no patient is left untreated.
Thank you so much. For question number 4, is tolerization permanent or are tolerization boosts likely necessary?
That's really a mystery and an important question. It really has to do with the technologies that are being tested now for their ability to retolerize the immune system can have different lifespans. Think about it this way. Most of us have had a smallpox vaccine or vaccines that we are told last our whole life. Those vaccines induce lifelong immune memory. Exactly the same idea is true for tolerization. Is it possible that we can use tolerizing technologies to induce lifelong immune tolerance? That's the goal. I would suspect that some technologies would need an occasional booster, and maybe, hopefully, some of them would not, but that remains to be seen.
Thank you so much. And lastly, how can healthcare providers learn more about NMOSD tolerization?
Well, I think excellent presentations that are often provided by the Guthy-Jackson Charitable Foundation and other types of organizations, providers looking at clinicaltrials.gov, providers who are interested in immune tolerance, keeping pace with the rapid literature that's being published in this area. These are all ways that healthcare providers can learn more about tolerization. I will also say that patients with NMO and MOGAD will probably help providers learn about tolerization because there is a strong interest in potential tolerization therapies among patient communities, including that which has been developed by the Guthy-Jackson Charitable Foundation.
Dr. Michael Yeaman, thank you so much for your presentation and insights into emerging tolerization technologies. It's a very exciting and important topic, and we look forward to hearing more.
Thanks, Megan.
We would like to remind everyone that this presentation qualifies as a continuing medical education activity. To receive credit, please visit the GJCF Education Center using the QR code shown here. Select the session of interest and register for that session. View the session and fulfill the CME requirements and receive the CME certification online. We have been delighted to offer presentations on various topics in the NMOSD Grand Rounds series. With this presentation, that concludes the NMOSD Grand Rounds series for 2025. This is an enduring program and each feature remains CME eligible for 1 year following its release date. You can access the NMOSD Grand Rounds sessions, as well as other educational events, presentations, and media using the Education Center QR code shown here. And lastly, we are greatly appreciative for the support for the NMOSD Grand Rounds series, including educational funding provided by Alexion AstraZeneca Rare Diseases and educational funding provided by Amgen. Thank you so much.